All right, a test cross. A test cross is if you have an individual, you can look at that individual's phenotype, but you might not know the underlying genotype of that individual, right? You could have an individual that's showing a dominant trait, but you don't know if it's dominant homozygous or if it's a heterozygote that just happens to have a dominant allele. Right? A test cross allows you to see the underlying genotype of that individual by looking at what the offspring had. Okay? So you cross an individual. This is an individual that you're looking for what is its genotype, what is its underlying genotype. You cross it with a homozygous recessive, right? If you have, this is a dual test, or a dihybrid, or a test cross for two traits. If you think about an individual that you're just thinking, okay, is it homozygous for the dominant trait or is it a heterozygous? If you cross it to a homozygous recessive individual, then the ratio of the offspring, if it's a heterozygote, is going to be 50-50, right? Because that homozygous recessive individual is only going to give the recessive allele. And if it's a heterozygote that you're looking at, the individual that you're concerned about, if it's a heterozygote, then 50% of the offspring are going to get the dominant allele, 50% are going to get the recessive allele, and so 50% of the offspring show the recessive trait. If it was a homozygous dominant, then 100% of the offspring are going to show the dominant trait. Okay? So a test cross is to a recessive individual, and basically that recessive individual is not, I mean, it's contributing to the genotype, but it's contributing in a recessive way. So the individual that you're interested in is going to be the one that determines what the trait is for the offspring, whether they got the dominant or the recessive. So here, this is a cross for a dihybrid individual, right? I've got an individual here who's got two genes, an A gene and a B gene. Here they're got a wild type dominant allele, okay? And what this test cross is doing is looking for, is there any linkage? Are these two genes close together or are they not? So the linkage, or I'm sorry, the test cross in this is trying to see, is there any relationship? Is there any linkage between A and B genes? Okay? So if you think we've got evidence for, maybe you see just, as you've been breeding some flies or something, you see the A and the B traits seem to go together. Like they seem to be showing up together. If you want to actually test whether or not they're linked, truly linked, or if it's just an abnormality, you've just happened to look at the ones that look like they're going together, you have to set up a test cross. Okay? So you've got your heterozygous individual, heterozygous for A, heterozygous for B, and you cross it with a homozygous recessive for all the traits. This, if we're just talking about a pea plant, then you can do this either way. The male or the female could be the tester. If you're doing it in flies, or if you're doing it in organisms that have a sex link, sex chromosomes, or some type of sexual dimorphism, especially in organisms that have the male having a Y chromosome. So in mammals, in flies, when you do a test cross, you want to test cross to always the tester to be a male. Because the male, then you know what his contribution was in the offspring. Because if it's a female, you know the male gave the X chromosome. If it's a male in the offspring, you know that the male gave the Y chromosome. So a test cross is always something that you know what the tester is going to give. So in flies, a test cross is always to a homozygous recessive male. And you always do the unknown in the female. Okay? For other organisms, that's not quite that important, but you want to know what the tester is giving so that you can, because what we're trying to do is analyze what happened in this individual, right, the one we're testing to. So if you do this cross, and the A and the B gene are not linked together, then the expected phenotypes that you're going to get out is just going to be determined by what the female gave. Okay? So here's the male contribution, the tester, recessive A, recessive B. The phenotype is going to be determined by what it got from the female. Okay? And so, So in an unlinked, if these were on different chromosomes, or if they were sufficiently far away from each other on the same chromosome, so that always a recombination event was going to happen. But let's think about the easiest circumstance. If they're on completely separate chromosomes, then the chance of the A plus allele going with the B, it's a 50-50 chance whether that A plus goes with a B plus or with a B recessive, because they're on different chromosomes. So when those chromosomes line up at the metaphase plate in meiosis, and wherever if we track where that A plus chromosome goes, it's a 50-50 chance that the B plus is going to go with it, or that the B's were flipped the other way, and that the B minus, the B recessive, goes with it. Okay, so 50-50 chance. The same with the other allele, right? If I'm looking at a B plus, 50-50 chance going with the A plus or the B, right? So all of the genotypes that the female can contribute are equally likely. A plus going with B plus, A plus being going with B, the recessive A going with the dominant B, the recessive A and the recessive B. All of those events are equally likely. Okay? So the phenotypic ratio you expect is 1 to 1 to 1 to 1. If you looked at 100 individuals, you would think 25, 25, 25, 25. Right? We just simplify that to the 1 to 1 to 1 to 1. That was what you would expect for non-linked test cross. Okay? Two traits not linked doing a test cross. This is the actual cross that it's a similar cross to the one that you're doing in flies. Okay? We're looking at gray body, which is the wild type, versus a black body, which is the recessive B. And we're looking at vestigial wing, which is the recessive, crinkly, misformed wings, and a wild type wing, normal fly wing. These are both both recessive mutations. They only show up in a homozygous recessive individual. Right? The dominant is a true dominant. Okay. So if I set up a pure breeding vestigial wing, so this individual here, and you've already started doing this in fly nomenclature, but this is always how flies go. You always show the two alleles that you have separated by a slash. Okay, that's indicating the two chromosomes, right? On one chromosome, you've got wild type allele. On the other chromosome, you've also got a wild type allele, okay, for that gene. Then you put a comma or a semicolon, indicating we're talking about a different gene now. And then the other trait. In this case, this is a homozygous recessive, okay, VG/VG, so it's got vestigial wings. I'm crossing that animal from a pure breeding with another animal from a pure breeding black-bodied, right? Homozygous recessive for black-bodied, wild type for vestigial. And so what I have now is this heterozygous at both traits, right? If I take that individual and do a test cross, so a test cross is to a male that's homozygous recessive for both. If I do that cross and actually then look at the phenotypes of the offspring, what you see is the phenotype's not showing 1 to 1 to 1, right? What I've got is the gray-bodied individual and vestigial wing showing up much more often. And the other one, the vestigial wing and black-bodied, showing up much more often. What this indicates is that they are not randomly segregating. They're not on separate chromosomes. These two traits are actually very close to each other on the chromosomes. And it will also tell you what the original chromosomes look like. This is what the ones that are much more common are indicating that the parental chromosomes, this is what the parental chromosomes look like. One of the parental chromosomes from this, so not from the tester, because the tester is basically blind, right? The homozygous recessive, you don't really know what's going on those chromosomes. But on the chromosomes here, the gray-bodied allele is sitting close to, on the same chromosome, the vestigial wing recessive allele. That's one of the chromosomes that the mother gave. The other chromosome that the mother could have given is the reciprocal, because remember, she was a heterozygote. So the other chromosome, she had a black-bodied allele next to a wild-type vestigial allele. So those two genes sit close to each other, and we can actually know which alleles were on which chromosomes, because those ones show up much more often. Right? Those must have been the way that her chromosomes were. Now, the types where you actually have the lower numbers, that indicates that a crossing over event happened between those two genes. Right? Because if her chromosome was the B gray-bodied, B plus gene on the vestigial, then to get the B to go with the wild-type vestigial, a crossing over event must have happened on that chromosome. Okay? That's a fairly rare event because these genes are fairly close to each other on the chromosome. So I've given a little bit of your experiment away in your fly experiment. You know that these two traits sit close together on the same chromosome. You will actually try and map and see how close together they are. Right? And I'm just using theoretical numbers here. You're going to have to generate your own numbers. So they're not assorting independently. Certain alleles are going with each other more often because they're sitting next to each other on the same chromosome. Yeah, Will?
Okay, so I just want to have a couple questions. So we use the homozygous recessive as a baseline? We always use a male?
Yeah, baseline, I guess, is a good way to describe it.
Just so that we can, because that means we can tell something about...
It's because all the phenotypes are going to be affected by what the female gives, right?
Okay.
If he's giving all recessives, then the phenotype is being determined by whether the female gives a dominant or a recessive trait, right?
Okay, that makes sense.
Yeah.
And so then what you're saying about the differing numbers, the lower numbers versus higher numbers, is that those high numbers, the 1200, the 1400, you can tell that those sit on the same chromosome?
Yeah.
Because they're more frequently present in the phenotype?
Yeah, they're more frequently present in the offspring. Yeah, saying they must be sitting close to each other, and which alleles are sitting close to each other you can tell, right? The wild type B and the mutant VG are really highly present, right? So those must be the two alleles that are sitting close together on one of the chromosomes. It's a diploid organism, so she has another chromosome, and since she's a heterozygote, the reverse is true on the other chromosome. That the mutant, or the mutant black-bodied allele is sitting next to the wild type allele on that other chromosome. Okay? So if you ever see those two traits, so that tells you what the parents are. Then these two must be recombinant events. And so that's showing you, well, B plus and VG are sitting next together on the same chromosome from this female. And so a crossover event must have happened to get that VG swapped to the other chromosome and the VG plus on that same chromosome so that they're two sitting together now. Right? So the low numbers indicate a crossing over event. The high numbers indicate no crossing over, same chromosome as the parent had, the female parent had. Yeah. We'll do Tamara and then Rachel, okay?
Okay, so if this wasn't the case, it would be the 1-to-1 to 1 ratio?
If they were on separate chromosomes, then you wouldn't see something being more dominant than the other. You'd see them all just roughly equal. Right? Yeah.
And does the fact that there are a few low numbers indicate that they're not like that close together?
Right. So if they were so close together that a recombination event never happened, then you would never see those phenotypes. You would never see the homozygous recessive or the homozygous dominant. Right? If those genes were like right on top of each other, or if the trait was coded for by the same gene or something like that, then it would be right on top of each other and you never see the recombination. If you do enough, even if they're really close to each other, you're going to see a couple of recombinations happen. But that's going to give you an estimate of how far they are away from each other, and that's the next step. But I just want you to see deviating from the normal ratios. Right? Yeah?
So if you're trying to visualize this on a genetic map, then so B plus and VG would be one chromosome, and then below that would be the other linked chromosome, the B
Yeah.
and VG plus.
I mean, you can just think of a line existing here that represents the chromosome, and that B gene is located right there, and on that same chromosome, the VG gene is located next to it.
But to show it as linked, then would you have to calculate the map distance between them? And in
Right.
that case, then where square with the VG, then would you just show 2 lines crossing over from the chromosome and then calculate map distance?
I mean, you would just indicate these are so far apart, and then you have to calculate the percentage of times that recombination events occur.
Okay.
That's going to be a map unit.
Would only be 1 map distance. It wouldn't be like
Yeah,
2
right. No, 2 genes, you can only have 1 map distance from them. Right. It's just the relationship for how far apart they are. Yeah.
But if you're given like a different trait, then
If you were given 3, yeah, then you can calculate the map distances between each of them. Yeah. Now, recognize you can, if you're linked, if you're on the same chromosome, then you have the possibility of being linked. Right? But if you're so far apart from each other on the same chromosome that every time meiosis happens, you're so far apart that a recombination event always happens at least once, then far things on a chromosome are going to act like they're on different chromosomes. Okay? So you can only be greater than 50%, or linked together by more than 50% likelihood, right? If it's a 50-50 chance, then it's a 50-50 chance, right? You can't get any further apart on a chromosome than 50-50 chance of recombining. So in map units, you can't be further away than 50 map units, right? That's just the top, right? 50 map units says you might as well be on separate chromosomes, okay? But if they're closer together than a 50-50 chance, then we'll represent the map units as 49 map units apart, right? That 49 map units says, well, I know you're on the same chromosome, right? But if you're 50 map units apart, maybe they're on the same chromosome, maybe they're on different chromosomes. You can't tell. They just might be too far apart. Okay.
Is the map unit distance the same for wild types and non-wild types?
It doesn't really matter,
Okay,
right? Map unit just says how far apart are the 2 loci, the 2 locations on the chromosome. It doesn't really matter if you're talking about a wild type allele or, I mean, you could map 2 mutant alleles from each other. Because we're talking about the location on the gene where those alleles are coded for, right? Now, usually we just do wild type because it's usually the dominant one and it's just easy to keep track of. It's the most common. Once you start working in double mutants and triple mutants, they start dying and you just don't get good ratios and stuff. So most of the time, if you're trying to think about the map unit between 2 things, you pick a wild type allele for each of those to help you map, right? But you don't have to. Okay. The chi-squared test, one of your homework questions has you do a chi-squared test. Basically, this is a statistical method to say whether or not the data that you're looking at actually represents the ratios that you're predicting to show up. Every time you actually cross flies and do it, you're not going to get perfect numbers that come out that conform to your really nice 9:3 to 3:1 ratios or 1:1 to 1. You're going to have some fluctuation in your data. The chi-squared test says, is that just fluctuation in your data, or is that really saying that genes are linked together if you're actually calculating the phenotypic ratios of the offspring? Okay. So we'll do a chi-squared test on this guy. This is the same ratios that we just saw from that vestigial wing guy, right? So these indicate the parental chromosomes. These indicate the recombinants. And you're asking the question, is this deviating from what we expected? Statistically relevant to saying, yes, they're actually linked together, okay? So in a chi-square test, you always say, well, I expect them, your expected is they're on separate chromosomes. And so what I would expect if they're on separate chromosomes is to be identical ratios of all the phenotypes, okay? And you say, is my data sufficiently far away from the expected to say that they are truly linked? They're truly on the same chromosome. And it's not just a random fluctuation in data that's making me think that they're linked, okay? So what you do is you take the observed and you add up the parental chromosomes. So these 2 would be representing the 2 parental chromosomes that the female could have given. So we add those up. And then you take the 2 recombinant instances. Both of those phenotypes indicate that there must have been recombination between the 2 chromosomes. You add those together, those are recombinants, okay? And then you add together the expectants, right? And that's just going to be, same, 50-50 chance, right? Then what you do is you just stick it into this formula. This is the chi-squared formula. So chi-squared is what's represented there. It's the sum of all of the, so it's d squared. D squared is the observed that you actually saw minus what you expected to see. So you take what you observed, subtract what you expected, square that, and then divide by the expected. And it's the sum. So you do this for both, right? You would say, what did I expect the parents to be? 1,600. So you take the observed parents. So you take 2712 minus 1618, square that, divide it by 1618, what you expected, and you're going to get a number. And then you're going to add that to the recombinants. How many recombinants did you expect? Take the observed, subtract the expected, square it, divide by that, add that to the previous number. So you'll take those two and sum them together. Okay? So chi-square test you could do for a lot of different traits. For this example, we're just doing, is it the parental or is it the recombinant? Okay? That's going to give you a chi-squared value. Okay? So the math's easy. What you get is 1476 for my made-up numbers here. Now, that doesn't tell you anything, right? 1496, what does that mean? You have to put it into a statistically relevant analysis, right? And so I'm not going to go through how people actually generated the chi-square matrix here, but this is the chi-squared matrix, all right? This is a statistical, just saying how probable is it that your data is truly what you expected. So if it's very improbable, that means it's not what you expected, and you should reject the theory that they are on different chromosomes. Okay? So basically, we're trying to prove ourselves wrong. Saying, we think they're on the same chromosome, but let's expect that they are on different chromosomes, and if our data doesn't align with that, we say it's a low probability that it aligns with that, and so we would say, yes, they're linked. So then you can do further analysis. Okay? So the way you do this is you've got a whole host of probabilities here, or a whole host of numbers, and these are linked up with probabilities, okay? And we have to determine what is called the degrees of freedom. The degrees of freedom in this case is going to be the two, talking about two alleles, right? Two traits. Two traits are what you have. How is it best to describe it? You've got two traits. One of them you have to keep constant, and the other one is free to vary, right? So if we're thinking about segregation of the two alleles, if you think about one of the alleles is moving on the chromosome, and we're saying, was there a recombination event between them? So we're saying, did the one that was there not a recombination event? Is that what happened, or was there another thing? You're varying it by only 1 degree, okay? So you just have to say, one thing I'm gonna keep constant, and then I'm looking, what is the variance on the other? Okay, so that is the degree of freedom. We've got 2 genes. We're keeping one of them constant, so you subtract by 1. We only have 1 degree of freedom in this case. Okay? I won't ask you anything more than just 1 degree of freedom, but you should understand that you could do this for multiple traits all at the same time. And I can say, what's the likelihood of these 4 traits coming together? You would say, okay, I got 4 traits. I have to keep one of them constant. There's 3 degrees of freedom, 3 ways it could vary. Okay? So our degree of freedom is 1. So you find your number on the, now, our number was 1,479. So it's a very, 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 I mean, the highest on this chart is 10, right? So we're way out over here. That means the probability of our data being due to just normal variance is just ridiculously low, right? The probability is less than 0.0001 chance that the data we saw actually reflects the hypothesis that they're on the same, or that they're on different chromosomes, right? Our data is skewed way to the linkage side, right? There's just no chance that you would ever see that ratio just by random chance, and they still be on different chromosomes, okay? So they are definitely linked, very high probability of linkage, okay? The cutoff for this is 0.05 or 5% chance. If your data is any more than a 5% chance of being true, then you probably want to do further tests, right? You'd say my data set is inadequate to explain this, and you would want to repeat your experiment. But if you'd say there's a less than 5% chance that this is close, I'm 95% sure that it's on the same chromosome, then you could go ahead and say, yeah, I'm pretty confident they're linked, and do your further experiments there. But if your chi-squared numbers come down in here, and you're like, well, there's a 10% chance that this actually could just be variance in my data, then you'd want to do the experiment again. Do the same experiment again, breed more flies, get more data, and do another chi-squared test. If you consistently come back and it's, you're 90% sure, well, then you could probably say, yeah, they're linked. But the chi-squared test is very conservative. It says you need more data unless you're 95% sure that they're really on the same chromosome. Yeah.
So we do the equation, we get our number.
Yeah.
And our number, if it's less than this way, 5%, then we can have reason
Yeah.
to accept that they're on the same chromosome?
So you would find your degree of freedom.
Right.
You find your number, or closest to your number. That's going to tell you the probability that your data set is due to just normal variance. That the hypothesis is true, that they're on different chromosomes, but you're just seeing a random variance, right? So if our rejection value is here, right? I have to be at least 5%, well, I like to think of it backwards, right? If it's a 5% probability that my data is just normal variance, then I'm 95% sure that it's not normal variance. It's true linkage that I'm looking at. Does that make sense?
Oh, sure. Okay.
Yeah.
So when you said
There's only a 5% chance that my data set actually is really consistent with them being on two different chromosomes. I'm 95% sure they're not on the same chromosome, or that they're not on different chromosomes, that they are linked. Right? So the cutoff is 5%. If you're on this side, you should repeat the experiment. If you're 5% or less, then you'd say, yeah, they're really on the same chromosome. They're not acting like they're independently sorting. Okay.
So 5% or less, they're on the same chromosome?
Well, no.
They're on different chromosomes?
The hypothesis is, right, the expected thing that we use is that they're on different chromosomes and that they're independently sorting, right? And this is a question of what is the likelihood your data is just normal variance, right? Is really supporting the hypothesis, but you're just seeing random variance. This is saying there is a very, 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 very low chance that you would ever see the data that you see and them still being on separate chromosomes. Okay.
And then so just talking about going the other way, so say you have a 20% chance, you're not saying that you failed to reject your hypothesis completely, or you just need to run more tests? How do
You would need to repeat it again. It's basically saying this data is inadequate to give me the confidence I want in order to say that they're really linked.
Okay.
Yeah. So you would just repeat the experiment. You'd breed more animals to try and buffer out that. I mean, if you only looked at 100 animals, then maybe you just randomly looked at the 100 that supported your hypothesis, right? So you'd say, well, okay, my variance, it might just be because of my sampling size. Maybe I need to look at 1,000 individuals. And by 1,000 individuals, it will have buffered out, and I'll actually see the ratios closer to the ratios that I actually expected. Okay. Yeah.
How do we get these probabilities again?
You just look on the chart.
Okay.
This is the standard chi-squared probability chart. And so you would just use this. This is, and I don't want to go into how they generated this, but yeah, we'll just take this as a given, all right? We'll trust the statisticians here. All right. Once you actually know, and you've got with confidence, that these two are really segregating together, the two genes really are close together on the chromosome, the natural question is, okay, how close are they? Right. So this is the idea of genetic mapping, trying to give us numbers to how closely linked these two things are. This is a genetic map. A genetic map is a qualitative measure. Now we can put some numbers to it but they're not actually units on the chromosome. We're not talking about how many base pairs apart they are on a chromosome. That would be called a physical map. A physical map says your gene is located at this nucleotide position and the next gene is located at this nucleotide position, and we physically can measure a distance in nucleotides. Right? A genetic map is how likely is a recombination event going to happen between the two genes. Okay? That's what we call a genetic map. It's based on inheritance patterns, not based on actual physical location on the chromosomes. Now, the two are kind of related, and we can get an idea. If we take a genetic map and say, how likely is it that recombination occurs, you can usually kind of map that onto a round figure of how many nucleotides they are apart. But recombination can vary. So there's certain regions of the genome that don't recombine very often. So if it's in a region of the genome that's not recombining very often, you can have things really far apart, but since recombination is unlikely, they will look like they are closer together, right? If there's an area in the genome where recombination happens a lot, you could have things fairly close together that look like they're on separate chromosomes. But it's just because recombination was more likely in that region. So you have to know something about the genome, and you have to know more to get a physical map. But for a genetic map, you can just take the numbers, and basically you just get a percentage. How often is a recombination event happening? Okay. So we've decided to keep our hypothesis, or I'm sorry, reject our hypothesis and say that these are on the same chromosome. And from our numbers, the parentals were the B plus allele and the VG allele were on one chromosome, next to each other on one chromosome. The B recessive and the VG plus were on the other chromosome. Okay. And what we're asking is, what is the likelihood that a recombination event happens between those? And so we already have the numbers to do this. We know what the parental numbers are. We know what the recombinants are. So you just take the total number of recombination events that happened. You divide by the total number of individuals you looked at, and you just get a percentage, right? You get a ratio there. This ratio is 0.16. That represents there is a 16% chance of recombination every time the female is undergoing meiosis. So when those chromosomes, those homologous chromosomes, line up in meiosis, there is a 16% chance that there's going to be a recombination event that happens between them. Okay? That is a map unit. The percent chance of recombination is just map units, 16 map units. It's also been called centimorgans after Thomas Hunt Morgan. So 16 map units, 16 centimorgans, 16% recombination. Those are all equivalent terms. Okay. Yeah.
Is it like an actual distance? Like when you put nucleic acids to it?
Well, if you knew something about what chromosome it is, you could probably estimate.
Oh, okay.
But I mean,
Is it just
recombination happens. It's a factor of the chromosome. It's about physical characteristics of the chromosome. Okay. And we'll talk about this when we talk about human genome mapping. Okay. Because, for instance, smaller chromosomes tend to have higher incidences of recombination. Because it's a smaller chromosome, the two chromosomes come together in meiosis, and even though it's a small chromosome, it's going to want to recombine. Okay? So recombination events and genes on small chromosomes have higher chances of higher chances of recombination happening. So they look more like they're spread apart. Right? Higher chance of recombination. They're close together on a small chromosome, but they act like they're on different chromosomes because a recombination event happens. If you've got a really big chromosome, things happen in a more standard way, right? So if you're talking about a small chromosome, if you're estimating how close it is, then you're going to be off if you're doing genetic maps trying to correlate that to a physical location.
So smaller chromosomes might have more map units per se?
Yeah, two genes on a small chromosome might be the same nucleotide different distance away from each other as two genes on a big chromosome, but they will have a bigger map unit. They will appear to be further apart from each other because recombination is happening more frequently, even though there's the same distance in nucleotides apart as the two genes on the big chromosome were. We'll talk about this more and why that's the case, but this is just a percentage game, right? Yeah.
So for the parental, you always only represent one chromosome from each parental. You don't write both chromosomes. Or so for instance, like the parent, like let's say that's the mother, then it will be B plus VG on one chromosome, the other one would still be B plus VG. But we all only write one.
I'm talking about what did the mother's original chromosomes look like. Her original chromosome had B plus and VG on the same. And then on the other chromosome, she had B, VG plus.
But isn't like when we have the B plus, and then VG, VG, and then crossed with the BB, VG, wouldn't the mother be B plus VG for, do you see what I mean? Like they're both B pluses. So where did the B come from?
So the original parental strain that we used,
Yeah.
B plus, so it had gray body, had vestigial wings. The other one had a black body and normal wings.
Yeah.
When you cross those, this is what you got. This is the mother's genotype. She's a heterozygote, right?
Oh, okay.
This is the mother that we're talking about.
Okay, so you're not talking about the mother.
No.
Not the grandmother, the mother.
Yes.
I was getting
Yeah.
confused there.
So what I'm doing here is I'm saying, so this is the mother that I then did a test crossed with, right? Okay. So this is how I represented her before. I was talking about her alleles with a semicolon. Now I'm representing by saying, okay, I actually know that that B plus is on the same chromosome as the VG. So this line is indicating these are on the same chromosome, line, these two were on the same chromosome.
Okay, but
Okay.
if the mother is B plus, and then on one chromosome, the B is on another chromosome, and the VG is on one, then why wouldn't it be B plus VG plus? You see what I mean? Like
No, I'm getting confused with the B's and the VGs. Well, come see me with your question afterwards, or we'll chart it out and we'll explain it. Okay. Because we need to talk about a 3-point test cross. It looks complicated. It's really straightforward. What you have to do, we're looking at 3 different genes now, right? And so there's going to be a map unit between, so 3 different genes on the chromosome. And we're not telling you which ones are which right now, but just think of 3 genes on the chromosome. There's going to be a map distance between this purple one and the green one, and there's going to be a map distance between the green one and the red one, okay? So if we're doing a 3-point test cross, we're looking at the distance between the middle and one end and the middle and the other end, okay? In this case, you have a yellow, elongated, dry fruit. Okay, this is wild-type fruit, so these are all dominant traits. But this is a heterozygote, okay? It's showing all dominant traits, but it's got a recessive allele hidden in the background, okay? We're going to do a test cross to a homozygous recessive for all of our traits. So this is a purple, round, juicy fruit. So PRJ indicates recessives. P plus, R plus, J plus, that indicates the dominant alleles. Okay? So we've got a heterozygote here, homozygous recessive. We're telling you that this is a heterozygous individual at each one, but you don't know the order that these genes sit on the chromosome, nor do you know which alleles are sitting on either of the chromosomes. Okay? So if I give you this genotype, that does not mean that the P plus, R plus, and J plus are on one chromosome and all the recessives are on the other. You could be mixed and matched, right, of where the alleles are. We don't know. So the way to tell where the alleles are and the order of the chromosome is to look at the test cross progeny, right? I'm going to test cross this to a homozygous recessive for all my traits, and then you just look at the ratios that come out. So I've got, since I've got 3 different genes, each of them have 2 different alleles, I've got 8 different phenotypes that I could see, okay? And on my next slide, I have these just bigger, okay? The first thing you do is you want to know, what did the original parent's chromosomes look like? Okay, we're going to forget about the test crosser, like that homozygous recessive. We don't care about that individual. What we're saying is, what did the parent that was the heterozygote actually look like? Okay, what did her chromosomes look like? The first thing you do is just identify the very most plentiful offspring. We kind of take it as standard that the recombination events are rare. So most of the time, the chromosomes are going to stay intact, and recombination is going to be fairly rare. Okay? So the 2 most common phenotypes are going to represent what the mother's chromosomal genotypes were. Okay? So wild type is the most common. Purple, round, and juicy is the most common. Okay? So we know that the genotypes for the mother that she most likely gives, most often gives, was all of the wild type alleles on one chromosome and all the recessive alleles on the other chromosome. Okay? Does that make sense? Her most frequent contribution is a chromosome that didn't undergo recombination. So the most frequent phenotype that shows up are the two most frequent phenotypes that show up represent the most common, or what her original uncrossed, unrecombined chromosome looked like. So one of the most common was all the dominants on one chromosome. The other was all the recessives on the other chromosome. So this is an easy example, right? We know that all the dominants are on one, all the recessives are on the other. The next thing you look for in a 3-point test cross is what is the rarest occurrence. The rarest occurrence is going to be where a recombination event happened between the first gene and the second gene, and a recombination event happened between the second gene and the third gene. That would be the most rare thing to happen is recombination between both, right? So that is down here. These two phenotypes, the yellow elongated and juicy, and the purple round dry, those are the most rare, you see? And so that must have been a double recombination event. That tells you what the order of the genes is, okay? Because look at the genotypes for this. Purple plus, round plus, but juicy is the new recessive trait that's showing up here. So there must have been 2 recombination events to give you that genotype. So the juicy allele, or the juicy gene, must be the one that's in the middle. Okay? There must have been a recombination event between P, the color, and juicy. So if we're thinking about going along the chromosome, we came across P wild type, P plus allele on this chromosome, and then it had to recombine to the other chromosome to get the juicy allele, the juicy mutant allele. And then it had to recombine back to the original chromosome to get that round allele. If you draw this out and actually show it, this is the only way it's possible. For this to be the rare event, the juicy gene has to be in the middle, because you had a wild type, and if it was an unrecombined, all of them would be wild type, right? But you get the P allele, then you had to cross over to the other chromosome to get juicy, and then you had to cross back to get the wild type again. So these two represent the most rare and will tell you the order of the chromosomes. Once you've identified what the order is and what the alleles are on the 2 chromosomes, then you just plug in the numbers of the recombinants to find the map units between these two. Okay? So if I want to find, if I know that P and J are next to each other, then I just look for instances where P and J are now going in the opposite arrangement. Okay? So I look, these are all recombinants down here. If I want to find the map distance between P and J, I just look for when P plus no longer goes with J plus. So P plus is going with the mutant J, so I'd use that number. And the reciprocal one you want as well, where the J plus is no longer going with the P plus. So you take those 2 numbers. Here, P and J are both wild type alleles, so don't use that number. There wasn't a recombination event that happened between those. Here is the recessive P and the recessive J. Those were on the original chromosome together, so this phenotype represents no recombination having happened between them. Okay? So you would take these 2 numbers, and you would take the double recombinants, because a recombination event happened there. And so you would add 46 to 52 to 4 to 2. You would divide by the total number that you looked for, and you've got now map units between P and between J. Yeah?
It should be 104 divided by 500.
It would be, is that 104? Yeah, 46, 52, 4, and 2. Yeah.
Okay. So can you go over one more time
Yeah.
You found, so you went to the rarest one, P plus, and then J, and you somehow made them next to each other. Tell me how you
Okay. So I know that these alleles are on the same chromosome, but I don't know what their order is. Okay? I know they were on the same chromosome. I know those alleles are on the other chromosome, but I don't know what their order is. But I know that those are the parentals, right? So you look for the most rare event. The most rare event means a recombination happened between whatever gene is in the first position and whatever happened in the second position, and a recombination happened between the second position and the third position one, okay? So you look for the rare phenotypes, that's these ones, and look to see which one is the new one, right? The original chromosome had P plus, R plus, and J plus, but this one is only recessive for J. So it must be that the chromosome had P plus, went to the other chromosome to get the mutant J, and then went back to the original chromosome to get the wild type R.
Yeah, that's where I'm being confused. I get the having to go to the other chromosome, but why couldn't it go P plus, R plus, then cross over to get the J?
Because that wouldn't have been a double recombination event.
It has to be double recombination event.
The rarest ones are the double recombination events.
Oh, okay. So that's maybe.
So it has to be true that a double recombination gave you this phenotype.
So then you have to figure out how that's possible. And
By putting them in the proper order, right? So the only way for a double recombination event to give you this genotype is if J is in the middle. Yeah.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.